should go forward according to, there we go. Okay. So the role of the environment in cancer, of course, has been investigated for a long, long time. And of course, when you look at the newspapers nowadays or go online, you're constantly seeing new things that somebody's come up with that look as if they could be causing cancer in some way. And the list is just increasing all the time. And the problem is that the data that are available on whether these things actually do cause cancer, or if they do, how they do it, we really know almost nothing about almost all of these factors. And they include things that we can't really do very much about. Endogenous processes, aging is a very important risk factor for cancer. Nothing much you can do about that. Obesity, inflammation, these are all endogenous processes that affect our cancer risk. And the statistical analysis is really very convincing on many of that. But there's other more controversial factors, like in the top left, the environmental sources. Very strong evidence for air pollution as a cancer risk factor. Microplastics is increasingly talked about as a major component in, for example, early onset colon cancer in young people. Microplastics is a candidate for involvement in that, but we don't have mechanistic evidence to demonstrate that really is the case. And of course, on the left side here, lifestyle factors. This is, of course, where we can most readily intervene as individuals to try and prevent our increasing risk in developing various types of cancers. We can control our exposures to some of these things, but where do you put your efforts and how much do you invest in these kinds of decisions? So the questions about what causes cancer and what you can do about it have been around for a long time. So this guy will be familiar to many of you. And he was talking about cancer, in 400 BC, so a long, long time ago. And the advice he gave at that point was along the bottom. If you understand ancient Greek, you'll be able to read this and tell me if that translation is correct. But basically, he said that if you give every individual the right amount of nourishment and exercise, not too little and not too much, you could find the safest way to health. And that's probably the right evidence for today as well. I mean, I think the depth of our knowledge doesn't go far beyond that. Do things in moderation, and above all, have exercise. For me, the single strongest factor that we can control ourselves, and it definitely has huge positive benefits in mice as well as in humans. So these are all things to think about. So to look at these questions, we have a grant that was funded by Cancer Research UK and the NCI 4 or 5 years ago. And that involves 9 teams in 5 different countries across the globe. And this is co-led by Nuria Lopez Vigas in Barcelona, Paul Brennan in Lyon at the IARC Institute. So the idea here is to collect data and samples from thousands of people all over the world, 26 different countries, I believe, and begin to look at not just the statistics of risk, but the molecular features of the cancers and the molecular features of the normal tissues in these individuals. Because if they've been exposed to something chronically over many years, that there should be signs, some sort of smoking gun and exposure in the normal tissue as well as in tumors. So this is a grant now that's going into its 5th year. And the origins of environment and cancer go back hundreds of years. But this is probably the most important one from our point of view. It was shown by Percival Pott in the 18th century, a surgeon in London, that chimney sweeps were constantly going up and down these chimneys. They get impregnated with soot and coal tar, and many of them developed scrotal cancer. That was an association, but the Yamagiwa and Ichikawa, swapped off there. Yeah, they did the the causative experiment. They took the same stuff that you can find in coal tar, and they treated, in their case, hares, rabbits on the ears with this stuff, and they induced tumors. And that is, of course, one of the classic links between environmental exposure and cancer. This is one of the ears of the rabbits. It's pickled and in a museum in Tokyo. So that model of painting the skin turned into one of the most widely used models for studying multi-stage carcinogenesis. 150 years after Yamagiwa, Balmain and Schubig used a similar model with coal tar, and they combined that with proton oil, which is an irritant. It's coal tar contains lots of mutagenic compounds. It's also a strong irritant, but they combined that with proton oil, which is an extract.
Dr. Balmain,
Sure.
One brief moment to make sure that people online can see your slides. We'll just share your slides for those online.
Okay. Okay. So the people online are
Yes,
the
now
last.
they can,
Okay.
yeah, they were not able to see the full slides.
Right. Okay. So the model we've been working with is based on these original studies from Balmain and Schubig and others. They came up with the first evidence that cancer develops actually in different operational stages. There were two major different steps involved in getting through to full-blown tumors. They dissected this mechanism very elegantly. They showed that the coal tar on its own does not give tumors. They showed that inflammation with protonol does not give tumors. But when you combine the one with the other, then the mice will get skin tumors after a few weeks. And those are benign skin tumors. Some of them progress. Eventually, the mice succumb to malignant carcinomas after a few months or up to a year. But they did one very important experiment.
Click on just click on this one. Here, I'll do it for you.
How do I go back?
Arrow.
Yeah.
Up arrow to go back.
Okay, no, that's
Great.
okay. Yeah, the experiment they did is shown in this slide. What they did is they showed that if you treat with mice with coal tar, the first step, then you can wait for lifetime of the mouse, and then start with protonol here. And everything that happened here in the normal one still happens here. So whatever the coal tar does is permanent. It doesn't go away during the life of the mice, and they can still very susceptible if you wait until they're very ancient geriatric mice. So that launched the whole field of tumor promotion. So this promotion stage is the rate limiting step in getting cancer, rather than this first stage. And we can come back to what the mechanisms are of each of those in a few minutes. But that launched this whole field of tumor promotion. And the 1980s was really the heyday of that field. And these are all meetings, the proceedings of meetings that took place in the 1980s. Well, loads of this, it's just a subset. And there's lots of nuggets of information in these books that never really made it into print. So it's a vast source of information on mechanisms, what we learned about promotion back in the 1980s, because it was considered by these people and by John Cairns, who was director of the Coldstream Harbor Lab. Way back then, in 1982, he had this statement summing up the role of promotion in cancer more general. If you want to prevent people from getting cancer, you should be directing your attention more to the promoters in the environment than to the initiators. Because it's only by removing the promoters that can hope to benefit everyone. And so that was a very clear statement that had a big impact. But after the 80s, as you can see here, these are the publications over time on promotion. And they peaked in the 1980s, and then they just tailed off. Everybody stopped working on promotion. But at the same time, they started working on oncogenes. And that includes my own lab. We did a lot of work on promotion back then, and then it tailed off. There's all the excitement about oncogenes, and everybody moved into that field. I mean, there were a few labs, quite a few labs that continued, diehards working on promotion, and thank goodness for that. They kept the field alive and provided a huge amount of information that we're still using now. So why did everybody switch to oncogenes? I mean, the links between genetics and environmental exposures are along the bottom here. So beginning of the 20th century, 1914, the links between chromosome and cancer became obvious just by looking at a microscope already. Muller showed that radiation, which changes the mutations, can also contribute to this. Discovery of oncogenes, Thomas and Bishop got the Nobel Prize in the 1970s. And then this was really transformative in the 80s. It was shown that you could take DNA from a tumor, make that DNA, 3 billion nucleotides in every cell, and a change of one nucleotide in that DNA could transform normal cells. That was a really amazing observation that transformed the field and started everybody, including my own lab, to work in this field. The links between DNA and carcinogen mutagens were done by the Millers and by Heidelberger and others. Our work in this area started in the early 80s. We looked at carcinogen-induced mutations in now skin tumors with the model that I've already shown you. And this is just a very brief summary of what happened there. Instead of using coal tar, which has got lots of different chemicals in it, hundreds of different nasty chemicals, it had been discovered in the interim that DMBA is a really potent initiator. And TPA is a very important promoter. So the purified molecules, so we could work with those rather than coal tar and propanol. And with the DMBA, we could show that it binds directly to the adenosine residues across the whole genome and causes these 8 key mutations. And this is a very strong activating mutation of the last gene, which drives cancers. And if we change the carcinogen, we change the mutation. So there's a direct mutational signature that's linked to that specific carcinogen. That means that if a tumor was induced by this carcinogen, we could sequence the DNA and say that's the carcinogen that was mutation, like a forensic test of what caused the cancer, what initiated. So if you change from DMBA to a different type of mutagen, like MNU, methylnitrosourea, it finds the G residue. So the mutation there is a G28 rather than a T, so that you can easily distinguish between these different types of mutagens. So we think that well, we know that was the initiating event, but so at the same time, you have all this work on promotion in the 80s, and then this new work showing that mutations are critical. So then that started this dichotomy of what are the real causes of cancer? Is it mutagens or is it the promoters? And some people thought and still think that the mutagens that cause mutations are the most important causative factors, based on the fact that these are dominant transforming genes. You put a mutation in that cell and you can make it behave like a cancer cell. Bob Weinberg's work, Mariana Barbasset's work, and others who looked at the genetics of cancer. It was so elegant, so beautiful that it's very convincing that that's what we removed are the mutagens. But others thought that the promoters and the inflammation were more important, and that includes work from, say, Beatrice Mintz and Bissell, who showed that the phenotype of normality is dominant over the phenotype of transformation. So if you fuse a normal cell with a tumor cell, you get a hybrid which behaves normally. And Beatrice Mills showed that if you take a mouse-derived carcinoma cell and inject that into the mouse blastocyst, it can contribute to the normal tissues of the mouse during mouse development. So what she was calling a tumor cell could behave normally. So putting the tumor cell in the right environment would switch its behavior completely. So this is very convincing evidence. I mean, there are issues about all of these experiments that one can debate and argue about, but this was the major dichotomy in the field is what are the rate limiting factors? And this debate is still going on. I mean, this is a review from 2025 suggesting that this is the end of the genetic paradigm of cancer. So really one pretty extreme view of what are the major causes and where we should be focusing our attention. So this has been quite acrimonious at times, interesting, and is continuing as we speak. So a few observations reignited this debate. This is one of the major ones. This is work from the Sanger Center, and in particular, Inigo Martin Perena, and many others there. This is a paper on esophagus from Philip Jones. And what these groups did was they looked at completely normal tissue, and they looked for the mutations in the normal tissue. So cancers come from mutant cells. We should find those mutant cells in the normal tissue before cancers get there. And so this is the esophagus of a 24 to 27-year-old male. And each of these little circles is a clone of mutant cells. So they're all small, and there are many of them. As in this case, males get older, the clones get bigger and fewer. So by the time someone's in their 70s, they have lots of clones, and some of them are very large. So when a clone gets larger, that's exactly what happens during cancer development. But these phenotypes are normal. Esophageal cancer is actually relatively rare in the general population. So, but this reminded lots of people that this threat is there, that these mutant cells are in our bodies, and this is happening in all of our tissues, even in young people. Your pancreas of 20-year-olds, people in their 20s, already has many cancer-causing mutations. The mutation that you find in most pancreatic cancers is KRAS G12D. That's already in all of us, and it's just sitting there, but it's normal. The pancreas isn't abnormal, it's still functioning completely normal. So something has to make some of these clones start to become cancers, and that the best candidate was exposure to a promoter. So more recently, since going forward a few decades, with all these new technologies, you can now do this kind of forensic analysis in much more detail than we could previously. So this is an analysis of known human carcinogens that have been tested by the National Toxicology Program. They have tested, I don't know, the numbers fluctuate depending on who's giving the information, but between 600 and 2,000. And this is a summary of the effects of 20 of these chemicals that are known or suspected human carcinogens. What do they do to cause cancer in animals? So they all cause tumors. Some tumors arise in the lungs, some in the liver, some in the kidney. What the y-axis here is the number of mutations you find in all of these tumors. So all of these little small blips down here, these are all tumors that can lead to the demise of these mice. So they're equally potent. These tumors over here have more mutations. But of the 20 chemicals tested, there are only 2 or 3 that did it, that induce more mutations. Most of the chemicals are innocuous. They seem innocuous in terms of mutations. They're very similar in mutation burden to the spontaneous tumors, several groups of spontaneous tumors embedded in here. So it looks like these chemicals that cause human cancer, they're kind of, in mice, they're expanding, they're accelerating the development of tumors from cells that already have the initiating mutations. We know the mutations are essential to cancer. You cannot get tumor without DNA-based mutations. They're necessary, but they're obviously not sufficient because they're everywhere. We have normal tissue, but exposure to some of these things can make one of these clones begin to grow in a more threatening way. So again, just quickly, this was a repeat of the original Bloom experiment, where they said that the initiation event is actually permanent and stays there for the whole life of the mice. So that was 1947. So we went back and repeated everything they did, and they were absolutely correct in what they said. We did the same experiment, took tumors and sequenced them, looked at the whole genome sequence of these tumors. And what you see here, this is the y-axis where the height of this purple bar here. This is the number of mutations that you find in these tumors induced by a single treatment with this carcinogen, the MBA. So that's the molecular signature of the MBA is this here. And you can see that some of the tumors have very few mutations, like this one here, very few, this is a lot. The record in our experiments was this one here. And this was from the delay experiment. This is a mouse that had been treated, and then we waited a year and then started the promotion. So this mouse on day one got 50,000 mutations. But that didn't do anything for most of the life of us. So I think Bernard should be absolutely correct. So now we're using these new approaches and technologies to kind of dissect out all of these different stages. So we know that the initiating mutations are there, we know they're permanent, but what stops them, what stimulates them to start growing, and how does that actually happen? So you can envisage, so this is the model that we're looking at. It's a normal young mouse. This is epithelium, could be any tissue, we use the skin. When we treat with DMBA, then we get these colored cells, which all have mutations. When we treat with a promoter, the idea is that some of these specific mutant cells, the purple one in this case, begin to expand and grow, and you get purple tumor. But if you change things around, for example, mutations occur spontaneously. You're all sitting in the room and your blood cells are dividing. You're all acquiring mutations, thousands of them, every minute or every hour. So this is ongoing all the time. And some spontaneous mutations may be just as dangerous or more dangerous than these induced mutations. And so here we've got a mouse that has developed, a few mutations. They could also respond to promoters, and you get a red tumor from the spontaneous mutation rather than a purple tumor up here. You can take it further and expose the mice to other factors like PFAS as a candidate, microplastics as a candidate, other factors that may cause clonal expansion. And that might give you then a different type of tumor that PFAS could expand the green cell and give you a green tumor. So these are all things that are ongoing at the moment, is surveying all these many chemicals that I showed in the first slide and trying to understand, can they do this? If they do this, then that might indicate that these are the types of promoters we should be focusing on. We don't have this kind of information for almost any of those factors on the first slide. We've got them for these tool compounds like TBA, known promoters, but we haven't been able to do this yet for all of these other factors. So how are we doing this? So this is one of the new technologies that I think has revolutionized the whole field of forensic science as it relates to environmental carcinogens in general. Take a piece of tissue from mice or humans, or cell from culture, or organoids, make some DNA. It's alternative sequencing, which is corrected, it is error corrected. So you sequence both strands, so you need to find the mutation in both strands in exactly the same place. Then you're convinced that it's real. So you can now find one cell in a million in normal tissue that has one single mutation, if you know what you're looking for. Sequence the depth of 30, 40, 50,000, and you can find these single cells. And then you can ask, are those single cells expanding in response to promoters? And this, for example, using this assay, this very rapid assay that can detect very rare mutations, we can now very quickly show that the DMBA, within a week of treating a mouse with DMBA, you can find that mutational thing. These are all individual mice. These are controls. This is the clock signature, famous SPS1 clock signature, the ticking time bomb that is present in everybody every time a cell divides, every day. You get more of these mutations. These are not induced by and exogenous chemicals, as you know. These ones are, so a single treatment with a mutagen gives us this in the case of the MBA and gives us this in the case of MNU. So but you can get these results, you know, a few days after the actual treatment without making tumors. But speeds everything up. But this is one of the more important kind of conceptual slides about this selective clonal expansion that we get. So this is using an assay from TwinStrand, the duplex sequencing company that developed these kits initially. And so each of these boxes here, maybe I'll use this here. Each of these boxes here is DNA from one single mouse. So here is a mouse that has a mutation in codon 12 of the KRAS gene. So across here you have NRAS, KRAS, and HRAS, which are the core members of the RAS family. And the mutations at codons 12, 13, and 61, these are the hotspot regions where mutation happens there. This is a cancer-causing type of mutation. So these 10 mice up here, these are all completely normal mice. So they already have those mutations at a very low level. One cell in a million, possibly, or 100,000 has that mutation. When we begin to, when we treat with DMBA, now we get all of these new mutations along the bottom here. This is codon 61 of RAS, and that's very specific for the DMBA, because that's where it mutates. So we see all these new mutations coming up with that single treatment with DMBA. But then when we treat with TPA, as well as the DMBA, we get these large pink circles, which these are clones that have expanded in response to the promoter treatment. So this is a short-term assay. It's 3 or 4 weeks, long before we have any, dangerous tumors coming up. And the biggest circle here is the HRAS Q61L mutation. This biggest circle has the mutant cells that go on to give tumors. So this is way before there are any tumors. So it's a very powerful assay for seeing this promoter can stimulate the development of more dangerous clones. One thing that's come out of this, which I think is relevant to the whole concept of prevention, is this one here. When we treat control mice just with TPA without any mutagen, then we can see that some of those spontaneous mutations also respond to the TPA. So, for example, these are all mice that didn't get any DMBA. They only got TPA, but you can see that many of them have these two circles. One of them is KRAS G12D, and the other one is KRAS G13R. So these are two cancer-causing mutations that are there spontaneously in the animals and in you. These ones also respond to a promoter, and we will expand. KRAS G12D is, of course, the major RAS activating mutation for pancreatic cancer in humans. So this raises all kinds of questions about why we see these spontaneous mutations in our normal tissues, because the same thing's happening in all of us. Interesting thing about these mutations is if you follow them over time, then you see different patterns coming up. So in mice that have not been treated with DMBA, that's this whole panel here, and this on the right are mice that were treated with DMBA. You see that the HRAS Q61L doesn't do very much without DMBA. With promotion, the KRAS G12D promotes. This is with 4 weeks of TPA down here. But if you continue on through here to 8 weeks of TPA, this mutation disappears. It just goes away. The KRAS G13R, which was the other spontaneous mutation, it increases over time. It increases up to 8 weeks, but then it disappears. If you use the DMBA and then the TPA, again, you have this theme that the KRAS G13R goes up and then goes way down, and it doesn't give you a tumor. This is a log scale here. So this is about a 10-fold decrease over 4 weeks. This mutation just goes away and it never gives the tumor. The HRAS is the one here. This is up 4 weeks, 8 weeks, and it keeps on going up and gives you the cancer. So we've got these different mutations in the same tissue that are kind of, they're going up and down according to which exposure they have. And that's, I think, a very important thing to think about when you're looking at how you might prevent the dangerous ones from going all the way through without making the other ones, without activating other spontaneous mutations. And this is one that I think this is, if anything, relevant to some of the prevention studies that that are ongoing and prominent, but also that other people may be thinking about. This is the green bars here. This is over all these different treatment groups. This is the basically the variant allele frequency, so the number of mutant cells essentially there. The green bar is the KRAS G13R mutation with 4 weeks of TPA. These are the controls. It goes up. But this means that there was 4 weeks of TPA and then 4 weeks of withdrawal from TPA. Take away the promoter. And this is a very consistent finding. KRAS G13R goes up again, even more, when you take away the promoter. And you see that again over here. This is with DMBA. The green one goes up, way up, if you treat with 4 weeks of TPA, and then you remove the TPA for another 4 weeks. So you get this big increase in this green set of mutations here. So it's as if this mutant cell is stopping the damage to the tissue. And this mutant cell is maybe in a stem cell that tries to repair that damage. And this is a concept that is not new. It's been suggested before that you could have different types of stem cells in the tissues, probably all tissues. And some of these are emergency stem cells that respond to damage immediately, and others are more responsible for restoring homeostasis, bringing everything back to normal. And you could have different stem cells that have these different types of properties. And this kind of analysis can enable us to begin to tease out these differences. So this is the KRAS G13R, which I mentioned. So this is a slide showing the number of tumors that mice get from these assays. So it's not expansion, it's the number of tumors. So the blue here are the standard models. The mice will get up to 20, 30, benign tumors, papillomas on their skin. And these blue bars here are just basically different time points and so on. But if we knock out the HRAS gene, remember most of these have got HRAS mutations, but we're seeing these strange KRAS mutations in the normal tissue, which are spontaneous, but they never give us tumors. When we knock out the HRAS gene now, we get very few tumors, but they have the KRAS mutations. And the mutations that they have in the papillomas, in this green papillomas, 30%, that the malignant carcinomas that these mice get, 80% so far, this is relatively small numbers, but 80% of those malignant tumors have the KRAS G13R mutation, the spontaneous one that comes from nowhere that we don't understand. So it's when you change the selective constraints, say we're taking out a member of the RAS family, the other member of the RAS family is compensating for the loss of the first one, and it activates that clone. That clone begins to grow, and it will eventually give tumors. We had this kind of result more than 10 years ago, and we never understood it because we didn't understand these clonal competition mechanisms. Now there's a glimmer of understanding coming through from that. So I think in thinking about prevention, these are all factors that we're going to have to begin to consider. There's this clonal dynamics, which clones are there, which ones expand, which ones contract, which are the dangerous ones, and which ones are innocuous and maybe even trying to repair the damaged tissue. We don't want to get rid of those. So the idea of if we could get rid of all mutant cells in our bodies, everything would be fine. That's not true, because some of our stem cells may be dependent in normal tissue on some of these mutations. Okay, so I'm going to. How much time do I have? How are we doing?
We're at 10:39, so we have 21.
Okay, so I just wanted to go over a couple of mechanistic questions and experiments that are ongoing to try and understand this, and then look at the consequences for this kind of group, which is how do you translate some of this information into prevention. Well, what I've been showing you is that this tumor promoting agent, TPA, it makes cells grow in vivo, causes proliferation, and we get this expansion of these cells with this particular type of cancer-causing mutation. But also what we know the TPA does, it causes inflammation. And you can see this here. This is the number of blood cells, leukocytes, that get into the skin after a single treatment with TPA. And there's lots of them. So TPA causes proliferation, but also you get huge inflammation. So what are the causes of this phenotype that we're seeing? Because you've got mutations to start with, and you've got inflammation. So what's the interplay? So we did this experiment, which I showed this yesterday in the talk. So anybody who's there will know what the answer is. But if you have, you're considering what is our immune system doing when all these mutations are swimming around. Our immune system, we always think, is designed to find new epitopes, new bits of protein that are antigenic and foreign, and identify those cells and kill them. So we had expected that if we take these mice here, NSG, these are totally immunodeficient mice, and we treat them with DNBA and TPA, we're going to get a ton of tumors, either because there are more mutations or because they're just allowed to expand and run wild. And in fact, when you do that, you find that the NSG mice get almost no tumors. They're virtually completely resistant to getting these tumors. And so why is that? So Andrea Cortabi in the lab did this experiment with an Eve Candaba. I've been working closely together on all of this subsequent data on mutations and inflammation, along with the rest of the group, of course. So we used this twin strand assay on the ordinary mice, the FAB mice, and on the immunodeficient mice. So when you treat them once with the carcinogen, then both mice strains get these T to A or A to T mutations, which I showed you earlier. So they get the mutations. So the fact that they don't get cancer isn't because they don't have any mutations. And if we sequence deeply, we can actually see the HRAS Q61L in the immunodeficient mice. This is the critical mutation that we get normally. We can see that in the mice that were treated with DMBA, but whereas the wild-type mice, they expand all these clones. Here's the HRAS Q61L mutation here. They expand all these clones. The NSG mice, we don't see that. So what that tells us is that the immune system is essential to get the tumors in the first place. It's playing a dual role. Some positive, some negative factors are causing this, and we're presently looking at this in great detail. We're doing this a number of different ways. I've shown you the immunodeficient mice. This is a study of two different chemicals, both of which cause inflammation. There's TPA, which you've seen. There's a different chemical, which chemically is quite similar to the TPA, but there's a different side chain up here. This is a chemical that's been known since the 1980s. It causes strong inflammation in the skin, but doesn't cause any tumors. It's remarkable. You can treat mice twice a week for 20 weeks. Just very small tumors, and they don't do very much. There aren't very many of them. And the RPA and the TPA, they both cause these kinds of dramatic changes in the skin. They both cause inflammation. You see that here. This is the hyperplasia, and this is the leukocyte infiltration, which is with both TPA and RPA. But with the RPA, you don't get the tumors. So it's very, very low down here, whereas the TPA gives you usual 20 to 30 papillomas per mouse. So now we know that the RPA, as we saw with the NSG mice compared to FEV mice, the TPA expands all these clones. The RPA does not expand all these clones, although it already has very strong inflammation. So there must be something different about the type of inflammation that RPA is causing compared to the type of inflammation that TPA is causing. The inflammation is essential to get the tumors from the NSG experiment. And we now know that two different chemicals, they each cause inflammation. Only one of them gives the tumors, the other one doesn't. So you can then do flow sorting, and this is what Andrea did together with the collaborator Jose Angel at UCSF. They used flow cytometry to dissect out which kinds of immune cells get into the skin and which kind of immune cells are actually causing these changes that I'm showing you. And what that showed is that when you do this with with TPA, with RPA, and you look at all the different kinds of immune cells, then you find that the TPA causes a significant increase in these cells. These are monocytes and neutrophils. The RPA doesn't do that. So of all the cell types that have been looked at, then this is a very good candidate population to think about. These myeloid cells are induced only by the chemical so far. We need to test a lot more, but only by the chemical that causes the promotion in just these cells. So what do we know about these cells? These are monocytes, neutrophils. There's a very well characterized set of pathways that are activated by these cells. Interleukin 1B is up at the top here, activates this type of T cell, and that produces cytokines, IL-22, 17A, 17F, and these appear to be causing myeloid cell recruitment. They encourage the myeloid cells to come in to the tissue, and they also stimulate the epithelial cells, the keratinocytes, to start growing. So it's the perfect storm. You know, these chemicals can do both of these things at the same time. There are antibodies that are now being tested against several of these components here. I'm not going to go into the details, but this provides, you know, what we think is a window into thinking about cancer prevention, because here we have a chemical or a pathway that's involved in inducing cells that bring in cytokines that stimulate epithelial cells that contain the mutations, along with the myeloid cells that are necessary to make those mutations expand. So theoretically, if we inhibit these pathways, we may be able to inhibit the early process. And these are ongoing experiments right now, but it's interesting that that pathway I showed you with IL-23, IL-17 is already in use in humans. There are these antibodies. You may recognize some of them. Skyrizi is probably the market leader in antibodies that are used to treat psoriasis patients, which are a strong inflammatory response. You know, autoimmune reaction that gives inflammation in skin and flags, and they respond very well to Skyrizi because IL-23 antibody that basically inhibits this particular pathway. Okay, so a number of reasons to think that there may be, you know, we can look forward to testing some of these types of interventions, but there are all kinds of caveats that we need to think about in deciding where to go with this, and including some failures, I think we have to say, about how prevention trials have worked and what they've demonstrated. There are data going back to 1972 or even before that, showing that retinoids, which many people put on their skin because it rejuvenates your skin. It's a developmental regulator that causes cell fate changes. And it was also shown in the 1970s to prevent these papillomas from growing. So if you treat with TPA and retinoids, then you get hardly any papillomas. So sounds good. So that went further and was shown also in other models that retinoids can prevent early stage tumors from growing. Even in humans, there were some trials that suggested that they may prevent head and neck cancers from growing. But then the big trials were done in humans, and this is thousands of patients. And what they showed was that people who were at risk of lung cancer because of prior smoking history, for example, they start getting these systemic retinoids. And the trial had to be stopped early, is my understanding, because there were more cancers coming up, more lung cancers in the patients that got the retinoids compared to the patients that didn't get the controls. So, you know, what was that? Well, actually, the same skin model I showed, I don't know if that's on here, Next one. That's this one here. So as these studies were going on in humans, it was also shown by Stu Jasper and a couple other groups, I think, that when you use this mouse skin model we've been talking about with retinoids, you do get fewer papillomas, which are benign tumors. But the ones that made it to that stage were very dangerous, so they all progress because progression. Nice. And so there was still, the thing worked to prevent the benign tumors, but it didn't prevent the dangerous ones, which is really what you have to think about preventing if you want to have this working in humans. So this was missed. If we'd known this before that human trial, that could have saved a lot of money and a few lives as well. So this field is moving now pretty rapidly, I think. This is some studies from Charlie Swanton and his team who really have contributed in a major way to advancing this whole idea of how you approach cancer prevention. So Charlie's group, they identified this paper here, which many of you have probably seen. They showed that, first of all, air pollution is a potent producer of lung cancer in humans. Acts mainly through EGF receptor mutations rather than through RAS mutations. It's more potent in females than in males, particularly Asian females. So a lot of interesting data on this model and the patients with cancer. What Charlie's team did, they identified in their mechanistic studies interleukin 1B as a candidate mediator of this promotion effect. And that was based on this observation here from a totally different human prevention trial, but in this case it was prevention of heart disease. So the individuals, it was 20,000 people who took part in the study. They were treated with this antibody, which was known to inhibit the effects of interleukin 1B. And that was predicted to prevent the development of these heart problems. It didn't actually do that, but what it did was prevent the development of lung cancer in these same patients. So that's a human epidemiology trial that worked. It wasn't designed to work that way, but it did work, and it gave significant evidence that inhibiting that pathway could prevent lung cancer. So that's, I think, a pretty much landmark kind of observation. And the pathway, of course, that interleukin 1B is tackling is this one here, which I showed you earlier. It's exactly the same pathway that we find these tumor promoters are activating this pathway. So there's a nice, confluence of these different approaches to looking at this problem. One could now begin to think about interleukin 1B pathway inhibitors, or even some of these other modules within this network of cytokines and the receptors. How do we intervene to possibly prevent some of these events from happening? This basically is the CanPath trial, which was the one that showed the effect on lung cancer. There are some hints now that these other pathway members, IL-23, IL-17, and psoriasis, there are some hints that they may also reduce the level of cancers of different types in the patients that have been treated. But this is a very open area. We don't have the data. There's a lot more data that would have to be mined to be able to do that. But this is something I've been speaking with about. If you want to do these large-scale prevention trials, how do you think about doing that? You just treat everybody. Ideally, yes. Everybody would know that they should take this pill every day and not prevent the cancer. Ideally, it's not going to happen. It's too expensive and far too dangerous. So you need to identify the people who are at the highest risk. And this is one way. It's a very interesting paper, also from the Swanton Group. What they did was use the data from the UK Biobank, who've been following, hundreds of thousands of people over a long time. There are blood samples taken at different intervals, and a company, I can't remember what the company was, but has analyzed the proteome in the blood serum of all of these people. So they came up with a predictor. Somebody gets lung cancer, after 10, 20, 30 years, what are the potential predictors of them developing lung cancer? And they found this set of 14 protein peptides in the serum that predict this individual's got a high level. This individual's going to get lung cancer in the next 5 years. And so this is now a very promising approach. Even if this doesn't work, I think this is the way you would have to go, is identify the individuals who are at the highest risk and treat them, because the treatment is going to be expensive. You need to convince a company to take that on and do these large-scale trials. So there are all kinds of logistic issues and problems in designing these types of trials. But I think this is beginning to show a route that's actionable and might even be realistic. So this is the last slide. So what we've talked about are how you might expand our knowledge of tumor promotion, which is still rudimentary, to ultimately trying to prevent development of cancer in the first place. So we have all these different systems up at the top, and the prominent team is exploring all of these. The duplex sequencing I mentioned, which is transformative, I think, for understanding the normal tissues, and really getting to the guts of what these clones are doing, these different clones that are talking to each other, competing with each other. How are they doing that? Because if you could intervene at this stage and prevent the dangerous clone, make the non-dangerous clone start to grow and take over, then that could contribute to preventing the emergence of this clone as a tumor. But we need to understand that competitive process in a lot more detail. All these fancy new techniques along the bottom, single cell profiling, analysis of epigenetics, lineage tracing in mice, are all being used and applied to these questions. What we're really hoping is that this year, at the end, of course, we have AI, which we think is going to solve all of our problems. And there are, I think, I'm very optimistic about that, rather than the doom and gloom kind of scenario. I think it's transformative, and at some point we may be able to take a look down the microscope and see a piece of tissue which is normal tissue, or it could be tumor tissue that either tells us what caused the tumor, or tells, in the case of normal tissue, what is the risk of that normal tissue of developing into a tumor. I think that will happen, but don't hold your breath. It's not going to happen tomorrow, but it's definitely something for the future. So this is the prominent team, and so Eve Kandava is here. She was first author of a paper that just appeared last week, which describes some of the work that I've been talking about. So if you missed it, then much of it's going to be in that paper. There's also a review that appeared a few months ago, which summarized some of these slides were in the review. So it's all there if you want to catch up online. So Eve has done much of the work on the clone expansion, the early clone expansion. Andrea is Italian postdoc in the lab, and has done a great job on really beginning to understand this, the immune involvement, immune components, and which pathways are involved. And this is the core team here. They've all really been essential to get this to happen. And of course, the CRUK, the Cancer Ground Challenges, was initiated by Cancer Research UK. It was also funded by the NCI. So a lot of funding and planning has gone into all of this. And so I'm going to stop there, and if there's time, I'll take questions.
I think we'll start with questions in the room. I don't know if there are questions.
I just asked everyone to share their questions for the Q&A button.
Regine, you want to start?
That's a question. I'll start with myself. This idea of using the mutation to pinpoint the environmental mutation, environmental mutagens or cultures, is really promising, and it's recently have shown that, for example, for that might be explaining something early on several colorectal cancer. So I'm just wondering, the bill now need to figure out, for example, when did that mutation start? And in the human, that's going to take a long time to do, right? Because you have to find children and figure out their cohorts and see what happens in a lifetime. Are there any ways to do this, like answer this question faster using the mouse models?
Can you just repeat a bridge version of the question?
Okay. Well, the question is related to recent data showing that a bacterium, Colobacter, is found in humans and has been linked, for example, to early onset colon cancer. So how might we be able to find evidence for that mutation or even test strategies
Yeah,
for prevention in the mice? That's
yes.
the question. Yeah. Well, I mean, the data on human are very compelling that, you know, this bacterium is there and it causes mutations. That's very clear. What is kind of less clear at this point is how the bacterial infection is related to the ultimate causal events in the cancer. So if the mutational signature of the Colobacter, so it causes, like the DMBA, it causes a certain type of mutation, SBS 88, I think it is, 89. So you can see, if you sequence somebody's colonic DNA, you can see that they've been infected with that bacterium. So that in itself is remarkable. So they've been exposed at some point in their lifetime. But it doesn't tell you what the function of that, what causal nature of that infection has been. There's some evidence that the type of mutation that the Colobacter causes can cause point mutations, but also some small deletions. These may be associated with deletion of a critical tumor suppressor gene in the colon. So there could be a mutational history linked to bacterial exposure, but that's not clear yet. But what we know that bacterium is going to do when it first comes in is going to cause inflammation. And it could be that what you're seeing in this mutational signature is a sign of exposure, which is very good that we can do that. But you know, it could be that the exposure has resulted in inflammation, which has caused promotion. So those are the two questions I think are not addressed yet. And you could theoretically begin to do this with mice. We haven't done that. I think there may be some other groups are trying to do that because you need to detect the mice with the same bacterium, look for the same mutational signature to show that it's there. I don't know if that's been done. I think it may have been looked at in organoids. So the answer to the question is I don't know. So it's a long way to say I don't know, but I think that's a very important area of debate. And that will get sorted out.
The CanPath cohort, we don't have ready access to fresh frozen tissue, but we have access to so are those considered good enough DNA quality? Could you do like the duplex sequencing you described?
Yes, you can do duplex sequencing on smaller amounts of DNA because you're sequencing small regions of the genome, you have small applicants. So you can do it. It's clearly not as efficient. And there are issues. So, for example, when we do the duplex sequencing on skin, we generally remove the cells that we know don't have the mutations. So we can make a suspension of the epithelial cells, take the DNA from those, and do the. That's the cleanest way to do it. You take a whole chunk of tissue that has fat cells and bits of stuff everywhere, but not as well as the critical mutant cells. You're diluting your signal, so you need more DNA, and also the quality of the DNA that you get from sampling is lower. So we have not done that actually, but I think other groups have begun to do that. And I think that's certainly feasible, but it may take a lot of optimizing to make it work properly.
I'd like to comment on the success story of the reduction in mortality in childhood leukemia over the last 50 years, and the remaining 15 or 20% are very resistant, and also solid tumors. Is this work likely to progress that further?
So the question is about the advances in treatment of childhood leukemias over the last 50 years. And if this work is likely to progress or improve those statistics. That's basically the question. Well, the advances in childhood leukemia, I'm by no means an expert on this, but I think they have come mainly from understanding the underlying genetic basis of those diseases and finding very good drugs that can, treat the tumor or kill the tumor cells, and that has given this huge, increase in survival and responses to those disease types. Childhood disease cancers in general are kind of a different beast, I think, because there may be some developmental germline type of effect, may also be an environmental effect, but that's very dubious. Again, it's not my specialist area, but my understanding is there's no clear environmental factor that causes childhood leukemia that you could look for using any of these technologies. It happens at certain frequency. The bad luck factor is there, but also germline predisposition that may conspire to get a particular type of genetic event, and the child can get leukemia. And the statistics are much better than they were 50 years ago, but they're still disappointing. Kids are still dying of some of these tragic. But I don't know that this particular technology will help directly to address that. Yeah.
Okay, so you started with what the Hippocrates said, right? Like informed BC. The other thing that he said was it doesn't really, what matters more is what type of person is getting the disease rather than what disease the person gets. So that's the concept of precision medicine.
Right.
In. So to achieve that, in a sense that you also bring up at the very end, is using a lot of the circulating signals to figure out what type of the high risk population, etc. And we do a lot of work on that, is to figure out who is at high risk using circulating tumor signals or proteins, etc. But the fact that we can also figure out their environmental exposure using their mutations. Currently, I don't see the two fields being sort of synergized in a way. It seems to be two very distinct fields. One is using more circulating signals to identify high risk, and the other one is using mutation epidemiology or mutations to identify environmental exposure. Do you see a way that we can be more efficient trying to bridge these two and then identify those people who can benefit?
Yeah. That's a really, really important question, actually. Because we know that different groups of individuals have different susceptibilities, and teasing out what's environmental, what's their local environment, where they live, and what's genetic. And because we inherit polymorphisms from our parents, obviously, and our germline has been filtered, and we are unique individuals with our own little, composition of alleles that make us who we are, and what we're susceptible to, what we're resistant to. And that's very, very difficult to tease those things out, even in the prominent cohorts where they have thousands of samples. Very difficult to see, if there's an area of the world where people are at very high risk of esophageal cancer, and those areas exist. The explanations could be, well, those people came from, if you look at their genetic history, they share alleles, so they're more susceptible because they have these alleles. But they could also be more susceptible because they're exposed to a mutagen or a promoter, right? And that could mean that these people are at high risk and these people are at low risk. So that has been a major feature in the prominent kind of studies. Also mutagraphs, which is the precursor to prominent. I was directly investigating those kinds of questions. And so far, they haven't found evidence for, looking at this difference between people. In East and West Africa, for example, they haven't found evidence for a mutagen that is only in the high risk population using mutational signatures, right? So they've looked for those. They've sequenced hundreds of tumors, for example, esophageal cancer from East and West Africa, where I think it's a tenfold difference in incidence, but the high risk area had the same tumors, same mutational signatures as the low-risk tumor. So there wasn't some new carcinogen, mutagen there that's doing it. It could be a new promoter there that's causing it. So these people are more exposed to a certain promoter than the other ones, and so they're for that reason at high risk. But it could also be what you're talking about, which is, are these people different to start with? Do they have is their genetic pool make them slightly more susceptible? I haven't found much support for that hypothesis at the moment, but I think it really needs a lot more detailed analysis. So, for example, in a recent study, again from the Sanger Center, they looked at kidney cancer from all over the world, and they found a mutational signature that was enriched in kidney cancers from Japan, right? I think that mutational signature is also found elsewhere in Asia, and also is found in the liver, not just the kidney. But there is a mutational signature in these people. But then the question is, are they exposed to something there, or are they genetically predisposed? And genetic predisposition, I don't think has been adequately looked at in the aftermath of those papers, because we know that the Japanese, for example, Japanese, also other East Asian countries, have certain polymorphisms that change their metabolic enzymes. They deal with carcinogens in slightly different ways. Just in the same way that there's a Japanese a signature for alcohol exposure, which is very high in Japan, because they have a polymorphism that they react very strongly related to metabolism of alcohol, the aldehyde pathway. And so they are more susceptible to alcohol in general. You get this flushing syndrome, and they also have more of this mutational signature from drinking alcohol. But you could say that that's because of their genetic predisposition in metabolizing the alcohol, rather than they drink a lot more alcohol than other people. You know what I mean? Yeah. And the same thing for these kidney cancers that have come up. There are polymorphisms that control, for example, the metabolism of haloalkanes, and these are different in East Asia from Western populations. They're also different in different mouse strains. So you can take two mouse strains, one of which has the susceptibility metabolism allele, and it will get more cancers from a certain chemical than the other strain. So I think it's a really important question. It needs a lot more effort to begin to understand that. And I think mouse models can dissect these things out in mice, right? We have many, many different strains. We know what they're susceptible to spontaneously. We know how many of them respond to carcinogens. What we don't know is, we don't have all this data, even for mice, what kinds of mutations they get. Is that strain dependent? How they respond to promoters? Is that strain dependent? We really don't know the answer.
I'm gonna ask, we're over time, but I'm gonna ask one question online, just to be fair to everybody. And it kind of relates to what you were talking about. So this idea of, the role of promotion in this and how we translate that into a public health message, like into the public, when you have high risk groups that need to be targeted for interventions, for example, to remove that promoter exposure. So how do you actually saying something like cigarettes cause lung cancer, smoking causes lung cancer is pretty straightforward, but then starting to think about how we broadly intervene on promoters and how we make that message.
Yeah, I mean, the advice, if you're trying to prevent cancer through the mutational route or through the promotion route, is different, very different, I think. I mean, clearly, the mutations are permanent, right? We're exposed to stuff all the time that gives us mutations. Once they're there, no way you can get rid of them. And it could be that if you find a genetic way, in the whole body to get rid of mutant cells, you kill people. If you're trying to do that, because those cells are, every cell in my esophagus probably is mutated, and so get rid of those cells, then that's not very good. If it's a promoter, there are various ways. Obviously, preventing by removing promoters is one thing. Air pollution. Good luck with that for the foreseeable future. But some of these things are potentially actionable. Understanding how promotion works may help. And if there's something that people could do immediately, is to look at these potential promoters and change your lifestyle. Each individual can do that. Otherwise, you're dependent on governments to change legislation. On pollution is going in the opposite direction in the US at the moment. People are removing restrictions on pollution in spite of all the evidence. So I think for promotion, understanding the process can help as an individual. For example, we know that promotion requires chronic inflammation. One dose of a promoter is not going to give you cancer. One dose of a mutagen is going to give you mutations that are there forever. But one dose of a promoter is going to cause some damage, inflammation, but it's going to subside. You need to get promotion to work. You need to, before the promotion subsides, from that first dose, you have to treat again. Right, so you've got a cumulative effect of the inflammation, which perturbs the system. And that's the deregulation that leads to the activation of the clonal expansion. So we know that if you treat the mice once a week, instead of twice a week, you give them the same dose, but you only do it once a week, they don't get the tumors. So the timing of exposure is absolutely critical. So if you drink every day, or if you smoke cigarettes every day, then you're getting the initiator dose with cigarettes, but you're also getting the promoter 10 times a day. And so you're doing the perfect experiment on yourself. But if with smoking, it's difficult to dissect out what the mutational versus the promotion effect. It does both. With alcohol, it's almost certainly a promoter. But maybe by cutting down the frequency or the dose. With promotion, the classic information that always works. You cut down the frequency and you cut down the dose. And that works to almost completely prevent cancer. Once a week for 40 weeks doesn't give tumors, whereas twice a week for 20 weeks, same total dose, gives loads of tumors. So somewhere in that spectrum would help. So just pushing out you're reducing the frequency and or reducing the dose. These are both things that everybody could do if they take the list seriously of what can be positive.
So much. Those are really fantastic thoughts. So please join us for coffee and pastries just around the corner in the main room out here, and we can have more time. So great. Thank you.
Yeah. Apparently, this is San Francisco Pride. So I'm not making a statement.